Greetings. This is Charles Darwin, and I'm here to talk to you today about bias and scientific experiments. Now, bias is something we usually say in a bad and negative way, as if, well, you're biased, and so somehow you're a bad person. But actually, everybody is biased. Every human being has biased observations. And when we do scientific research, we have to avoid that bias. Therefore, we have to take special measures to keep the minds of scientists and of the participants in the study that the scientists are running from having bias influence their observations and their measurements. And we have to do that. We have to set that up in our experimental design precisely because scientists, like everybody else, have biases. It's not that scientists are less biased, it's that scientists take deliberate steps to avoid the bias that they know we all have. Now, one of the things that some food scientists and marketers also would like to know is consumer preferences for different kinds of tomatoes. Here we have some yellow cherry tomatoes, here's some red cherry tomatoes, and some of the red cherry tomatoes actually have a little bit of blue color in them that makes them look purplish. Okay, so we have three different colors of tomatoes. And naturally, a marketer or a food scientist might want to know which one do people prefer. Now, that doesn't mean that one of these is better than the other, but what they really want to know is which one do people, for whatever reason, prefer. But you see, you would think it would be perfectly obvious to do this. Just take one of these red tomatoes, and start eating them and say, yeah, it's pretty good. And the yellow tomato, and yeah, that's pretty good too, but not quite as good. Now, why did I say that? Well, one of the reasons I said that is because I might really have preferred the flavor of the red ones to the yellow ones. On the other hand, we have in our minds a bias that tells us that tomatoes are supposed to be red, therefore the redder a tomato is, the better it is likely to taste. Now, the only way to get around that particular problem is to try to remove the sensation that is biasing the results. In this case, sight. So what food scientists may do is they may use blindfolds. There's other things that they can use as well. Now, I've got here three different kinds of tomatoes. Let's see, here they are. Yeah, okay. And so I'm going to start moving them around, and I don't know which one to switch now. I'm going to put them back, and now I'm going to try one of each and see if I can tell you which one I think is the best. My bias is to say that the red one is the best. Let's see if that happens. Okay, that's number one. That's number two. And that's number three. Okay, now I can't really tell the difference between one and two just on the basis of taste. So I suspect that one of these is red and the other one's yellow, and that this one's probably the purple one, although I don't really know that, do I? Now there's another factor that's involved here. Oh, let's see how this turned out. Oh, I actually thought this was the red one, but I got it correct. One was red and one was yellow, and this was one I did not prefer. There's another factor at work here, and that's the sequence effect. Not just participant bias of seeing color and interpreting it as flavor, but sequence effect. It's possible that I would like the best the first tomato that I tasted. Now, that's not likely to be the case here because I stuffed a couple of tomatoes in my mouth before I started the comparison of these three, but that's still possible that you will like the first one better than the second one, no matter what kind it is and no matter what color it is. All of these are things that scientists have to address in their experimental design to make sure that these kinds of biases, inevitable as they are, do not influence the results. This is Charles Darwin. Tally ho, and amen.